Swayam Prabha. Digital India. Educated India.
Welcome to this 7th lecture in this course on eukaryotic gene expression. Today we are going to talk about the role of chromatin in the regulation of eukaryotic gene expression. Just to recapitulate what we have been discussing the last few classes, we began our discussion with eukaryotic RNA polymerases. We then went ahead to discuss about the core promoter elements around which the pre-initiation complex is assembled, which consists of general transcription factors and RNA polymerase II. And then we discussed about the various general transcription factors and how they function. And then we went on and then discussed, in addition to the core promoter elements, there are also many upstream elements which play a very important role in the activation of gene expression in eukaryotes. These can be proximal promoter elements, distal promoter elements, enhancer elements, silencers, and so on and so forth. Then we discussed the actual transcription activation takes place by transcription factors, sequence specific transcription factors, which go and bind to some of these enhancer sequences, and binding of these transcription factors to these enhancer sequences then enhances the rate of recruitment of RNA polymerase at the core promoter region. This is how enhancement of transcription takes place in eukaryotic cells. We also discussed very briefly the structure of the various transcription factors, what kind of DNA binding domains these transcription factors contain, what kind of transcription activation domains these transcription factors contain, and so on and so forth. Now the major mechanism by which these transcription factors regulate gene expression is when they bind to the sequence elements, to the specific sequences in the promoter regions, they somehow have to interact with the general transcription machinery, and this interaction what facilitates faster recruitment of RNA polymerase, leading to enhancement of transcription. So in the last class I mentioned to you, one of the major mechanisms by which these transcription factors do this is by recruiting what are called as co-activators. It is these co-activators which serve as a bridge between transcriptional activators on one hand and general transcription factors on the other hand. And I told you in the last class that we will discuss about co-activators and what exactly is their role in regulation of gene expression. But what I will do today is, before I discuss with you about the role of co-activators, I thought it is important for us to now to understand the role of chromatin. So what I will do today is to really see what is the link between chromatin structure and gene regulation. You see, so far all our discussion, the last 6 or 7 classes has been about how the core promoter elements are organized and how the RNA polymerase and general transcription factors are assembled on the core promoter, and how transcription factors go and bind to upstream sequences, and how binding of these can result in the transcription activation. And we also discussed how various promoter elements can be mapped by techniques such as DNase I footprinting, gel mobility shift assay, etc., and how we can actually use either transfection assays in cell lines or cell-free transcription systems to study various promoter elements. But one thing we have not discussed so far is, in vivo, in reality, the eukaryotic cells, the DNA is not naked. DNA is actually wrapped around histones in the form and is organized in the form of nucleus. So this is a cartoon here shown here. In eukaryote, the cellular DNA is tightly wrapped up around histones and is organized in the form of nucleosomes. So there is about 147 base pairs of DNA which is wrapped around each nucleosome core particle, and the DNA gets nicely rounded around, and each this DNA plus the histone octamer, that is what is called, forms a nucleosome. So the one thing that we have to now discuss is, how do transcription factors gain access to the promoter DNA? Now, so remember, so far we have not brought this issue at all. We discussed so far as if the DNA is naked and the DNA sequences are freely available, so that the TF2D can go and recognize the Tata box or other core promoter elements, and transcription factors can go and bind to the upstream sequences, and then they interact and it can result in transcription activation. But this is not the case in reality. In reality, the DNA is organized in the form of nucleosomes, and that is what we call as a chromatin. So transcription activation actually has to take place not on naked DNA, but it has to happen on chromatin templates, which means the histones which are wrapped around the DNA and form the nucleosomes, these histones have to be displaced from the promoter in order for transcription initiation to take place. So, this is what we are going to next discuss in the next few classes. How do these transcription factors displace histones from the promoter region so that the pre-initiation complex can be assembled and transcription initiation can take place. There is a very nice history, so I would like to take you through a very brief history of how chromatin got involved in gene regulation. Now, for a long time, people who have been studying chromatin structure are a different kind of a group, and people who have been studying gene regulation were considered a different kind of a group. They never thought they actually have something in common. Many times people thought the only function of histones is to compact DNA, package DNA in the form of chromatin and chromosomes, and it only has a structural role. But it soon became very clear that histones may have many other role other than a structural role. In fact, it may have a very important functional role in gene regulation. Let us now discuss some of these issues in detail now. So, I told you the nucleosome core particle, which consists of core histones, what are called as H2A, H2B, H3 and H4. These 4 histones, 2 subunits each, together form what is called a histone octamer, and the DNA is now wrapped around this kind of end. So, the DNA is arranged in the form what is called as a beads on a string. And in between the 2 nucleosomes, you have what is called a H1 histone, which acts as a linker histone. And what is very important for our gene regulation is that when these histones form the histone octamers, you can see there are here thread-like structures which are depicted here. These are nothing but the N-terminal and C-terminal chains of each one of these histones. So, you have the histone octamer, but the N-termini and C-termini of each of these histones are actually hanging around. Okay. And this N-termini and C-termini are now going to have a very important role in the next few minutes, and these N and C termini are the ones which are now available for what are called as post translational modifications, and these post translational modification of the N and C termini of histones play a very important role in gene regulation. This is what we are going to discuss about in detail today. So, remember, because the DNA is tightly wrapped around histones and the DNA is organized in the form of chromatin, the chromatin actually acts as a general repressor of transcription, because DNA is very highly nicely packaged with these histones, and therefore the chromatin acts as a repressor of transcription. So, if transcription initiation has to happen, this DNA which is now wrapped nicely around the histones, it has to be unwrapped first. So, this is the first step that has to happen. And as people started looking at the chromatin structure and gene regulation, these 2 groups are working in parallel for a long period of time. Circumstantial evidences clearly indicated that chromatin may have a very important role in gene regulation. One such circumstantial evidence is cytologists and cytogeneticists who actually are observing on organization of chromosomes or organization of chromatin by using a number of staining techniques. As they realize that when they stain the eukaryotic nuclei or even the Drosophila polytene chromosomes or Lamb-Bruch chromosomes, etc., they realize there are some regions of DNA which get densely stained and some regions of DNA or chromatin which gets very thinly stained. So, these densely stained regions of chromatin are often referred to as the heterochromatin, whereas the lightly stained regions of nuclei or the chromatin is referred to as the euchromatin. And people soon realized that if you look at the transcriptional activity in the euchromatin and heterochromatin regions, most of the transcriptional activity is present in the euchromatin region, and most of the DNA which is localized in the heterochromatin region is transcriptionally silent. So, these kinds of observations clearly indicated that the organization of a DNA into chromatin has a very important role in gene regulation. So, if the genes have to be actively transcribed, they have to be in the form of euchromatin, and if the DNA is very tightly packaged in the form of heterochromatin, then the probability that genes in that region are actively transcribed are very, very low. So, these kinds of evidences clearly indicate that there is some link between chromatin and gene expression. So, the densely packed chromatin will block access of RNA polymerase II to general transcription factors to core promoters, and that is why probably the genes which are localized in the heterochromatin regions are not actively transcribed. So, if these genes have to be transcribed, these promoters of genes which are present in heterochromatin regions has now been converted into euchromatin. That means the histones have to be removed and the chromatin has to loosen, then only the proteins involved in gene regulation can go and bind to the promoter sequence and activate transcription. So, the chromatin in these regions, promoter regions, need to be unpacked, unwrapped or opened up before transcription initiation has to happen. Okay. So, the important point that we are going to discuss today is that if transcription initiation has to happen in eukaryotic nuclei, two things have to happen. The first step is the chromatin structure should be modified such that the RNA polymerase II machinery can now gain access to the promoter region. Okay. And then the next step is the actual recruitment of RNA polymerase II machinery to the transcription start site. So, it is like if somebody wants to perform either a dance or perform music. Now, the first stage has to be clear and the stage has to be set. Only then the people can come in and then make their performance. The same way, the chromatin, if it is out that if the histones pack the DNA tightly around and are in the form of heterochromatin, then there is no place for the players to come in and act. So, the first thing that has to happen is the stage has to be clear, that means the histones have to be removed and the promoter DNA has to be made available, so that then these general transcription factors or other transcription activities can come and then perform their job. So, the transcription initiation probably involves two steps. The first is modification of the chromatin structure to facilitate the various factors to gain access, so that they can come and then recognize their sequences and initiate transcription. Remember, these two steps are very important for all our future discussions now. Modification of chromatin structure is very essential for the regulation of gene expression in eukaryotes. This is the most important point that we are going to discuss today. Just like I told you, the euchromatin heterochromatin studies clearly indicated that the chromatin structure has a role in gene regulation. The same way, cell-free transcription studies also gave some clues that chromatin structure plays a very important role in regulation of gene expression. For example, we discussed in the last classes the way the cell-free transcription studies were performed. Initially, people started using crude nuclear extracts in which templates can be put in and their transcription activity can be assayed. Then once people started purifying general transcription factors, purified the RNA polymerase II and transcription activators, people started developing what are called as reconstituted systems, wherein you add individual purified components to the cell-free transcription systems and then add your DNA template and assay your promoter activity. But then as people started cloning many of these general transcription factors like the components of TF2D, TF2B, TF2A and so on and so forth, people started adding the recombinant general transcription factor to the cell-free systems and then started seeing whether you can demonstrate activation of transcription from these DNA templates in vitro. Now, very importantly, all the initial studies on this gene regulation were all again performed in the naked DNA templates. This is what we actually discussed so far. That means you take a DNA, promoter DNA, you hook into either a G-free cassette or something like that, and then add your nucleotides and add either clone transcription factors or purified general transcription factors, add RNA polymerase II, add nucleotides, one of which is radio labeled, and then you measure the promoter activity. People started realizing that naked DNA templates may not really reflect the real situation that is happening in the in vivo systems. People started using what are called as chromatin templates. That is, people who are studying chromatin structure and chromatin organization, so on and so forth, have developed protocols for purifying various histones like H1, H2A, H2B, H4 and so on. So, people took all these purified histones and then add them to the naked DNA, so that you can actually assemble these nucleosomes and you can actually prepare what are called as chromatin templates, in which you actually have nucleosome structures. So, there was a paradigm shift in the thinking of people working on gene regulation, that if you really want to interpret the results of the cell-free transcription studies to what actually is happening in vivo, naked DNA templates may not be the right way of doing experiments. You actually have to demonstrate how the transcription factors assemble on chromatin templates and how transcription activators actually activate transcription on chromatin templates. So, people who are actually doing all these experiments in naked DNA templates switch to what is called as chromatin templates, where you take your plasmid DNA templates containing a promoter and a reporter and then actually add histones, so that it forms a chromatin template, and then on this chromatin template you now add RNA polymerase, general transcription factors, and see whether you can now demonstrate transcription activation in a cell-free system. Now, when these studies were done, a surprising result that came out, that many of the activator dependent transcriptions could not be demonstrated when chromatin templates are actually used. In fact, very surprisingly, the Tata binding protein could not really bind the chromatin templates on its own. Okay. So, very clearly saying that when you have when DNA is packaged in the form of nucleosomes, or when you have chromatin templates, somehow these nucleosomes prevent the binding of TBP to the core promoter elements in vitro. Okay. So, somehow it became very clear, in order for the TBP to recognize the core promoter elements, you require transcriptional activators. Okay. So, there was the thinking change indicate that one of the mechanisms by which the transcription factorators may be facilitating the assembly of the general transcription machinery in the transcription site is actually by removing the nucleosomes or removing the histones in and out of the region, so that the general transcription factors can go and bind and activate transcription. So, chromatin templates need to be modified in order for the transcription factors to bind. This was the most important observation that came out of some of these early studies. Okay, clearly indicating that there is, you need to now demonstrate how actually what is the role of chromatin in the regulation of gene expression. But the question is, how do these chromatin templates function? How are transcription factors recruited on chromatin templates? A very important outcome of many of the number of years of research that came out is that the chromatin templates are actually modified and the general transcription factors are actually brought to the core promoter elements, and one of the important mechanisms by which this is brought about is actually by covalent modifications of the histone tails. Now, I told you in the nucleosomes, which contains the H2A, H2B, H3 and H4, the amino and carboxyl terminals of the histones, they will be actually, they are actually free, they are actually hanging around, and they are actually therefore amenable for post translational modifications. Okay, like phosphorylation, acetylation, deacetylation and so on and so forth. So, it soon became clear covalent modifications of the histones may actually have a very important role in the removal of histones or in the tight packaging of histones in the promoter regions, and this may have a very important role in the regulation of gene expression. Now, let us see how actually this was discovered. So, a number of studies, people who are actually looking at histones started looking, and then people realized that histones actually undergo a number of post-translational modifications. These include acetylation, methylation, phosphorylation, ubiquitinylation, sumylation and ADP ribosylation, and there are many other modifications that happen to the histones, just like for any other proteins. So, just like many proteins are subject to post-translational modifications, histones are also subjected to a number of post-translational modifications. The question is, what is the relevance of these post-translational modifications? Or these post-translational modifications affect only the chromatin structure, or they may also have a functional role. This is what was investigated. Let us now go one by one and then really see what is the effect of some of these post-translational modifications on histones, and let me begin with what will be the effect of acetylation of histones on gene regulation. Now, histones, we all know, are basic proteins. They are highly enriched in basic amino acids, especially the lysine and arginine residues. That's why they are all positively charged, and that's why they can very tightly go and bind to DNA. Because DNA is negatively charged, histones are positively charged, therefore the interactions can be very strong. What became very clear is the lysine residues in the N-terminal region of histones H3 and H4 can actually undergo acetylation in a reversible manner. Remember, whenever we talk about regulation in biological systems, most of these modifications have to happen in a reversible manner, then only regulation can take place. So, most of the changes that we see in biological systems are often reversible. When you add a phosphate, you also can remove the phosphate. When you add an acetyl group, you will also be able to remove acetyl groups. So, there are enzymes which may add acetyl groups, then they also have an enzyme which will remove the acetyl groups, then only the on off mechanism will actually work. So, an important observation that became evident is that the lysine residues in the N-terminal regions of the nucleosome core particle, especially which contains of H3 and H4, they can actually undergo acetylation in a reversible manner. So, acetylation of epsilon amino groups in the lysines result in reduced positive charge and therefore weakens the histone interaction with DNA. As I told you, one of the key mechanisms by which histones interact with DNA is histones are positively charged, DNA is negatively charged, and by acetylating these N-terminal tails of histones, you are actually bringing up additional negative charge, therefore you are decreasing the overall positive charge, and therefore the interaction between histones and DNA weakens, and therefore it may result in loosening of the histones in the chromatin region. So acetylation changes the conformation of nucleosomes and destabilizes the internucleosomal contacts. And acetylation of histones actually may alter interaction with other regulatory proteins. So these are some of the key observations that came out when people started looking at post-translational modifications of histones and their relevance to gene regulation. And this cartoon just tells you this is the lysine residue of a histone, which is shown here, and this is how the acetyl group is actually added, resulting in the acetylation of the histones. So now you can really see, people went back to see how exactly you can correlate these observations to some of the observations they made earlier. Now I told you earlier, heterochromatin, which is a highly condensed chromatin, is transcriptionally silent, and euchromatin, which is kind of a lightly stained regions of chromatin, is transcriptionally active. And when they actually looked at the acetylation of histones in euchromatin regions and heterochromatin regions, they in fact found acetylated histones are actually enriched in the transcribed chromatin or in the euchromatin regions of nuclei, whereas deacetylated histones are actually enriched in the heterochromatin regions or the densely packed regions of chromatin, clearly indicating that there is a correlation between transcriptional activity and acetylation of histones. So acetylation of histones somehow potentiates transcription by making nucleosomal DNA more accessible for transcription factor binding. This was some of the important outcomes that came by study of acetylation of histones. So now the question comes, okay, you found that histones are actually exist as acetylated proteins, and acetylated histones are actually present at the euchromatin regions of the euchromatin regions in the nuclei. But then the question comes, what are the proteins or what are the enzymes which add this acetyl group to histones? So you need to identify the mechanism by which histones are actually getting acetylated. And a very important, very surprising finding that actually came from some of the studies is many proteins involved in transcription factor activation, such as transcription factors or coactivators or even the TBP associated factors or TAFs, they actually contain histone acetyl transferase activity. This was one of the most important outcomes of all these the convergence of people studying histones and people studying gene regulation, that an important observation that came of this convergence of these two groups is that many transcription factors actually can serve as enzymes. And what is the enzyme activity? Their enzyme activity is their ability to acetylate histones. So you can see this I would actually call is one of the most significant results in the chromatin biology and in the regulation of gene expression. Identification of histone modifying activities or enzymatic activities in the transcription factors. So we are now going to use the terminology called HAT, which actually means histone acetyl transferases. So let us now examine some of the important observations that led to this conclusion. The first and foremost observation that a transcription factor actually possesses a histone acetyl transferase activity came from a transcriptional activator called GCN5 protein, which is a very well known transcriptional activator in yeast cells. Okay. So and this GCN5P actually was shown to contain histone acetyl transferase activity. This was one of the most significant observations in the area of eukaryotic gene regulation. And soon they realized that the human homologue of the GCN5P also has histone acetyl transferase activity, indicating that this enzymatic activity is conserved from yeast to humans. Okay. So for the first time, researchers working on histones and chromatin structure both got interested in transcription regulation. Remember, till then people who have been studying histone structure, chromatin structure were considered as a different group, and people who are studying gene regulation, gene expression were considered a different group. But for the first time they realized that they have to now work together, because the chromatin seems to be playing not only in compaction of DNA and have a structural role, it also seems to be playing a very important functional and regulatory role in the regulation of gene expression. So people who are working on chromatin structure and histones got interested in gene regulation. Similarly, researchers working on transcription regulation realized that they cannot ignore chromatin structure any longer. They have to come together and start working together if they really want to understand the mechanism by which eukaryotic gene expression is getting regulated. So the GCN5, the yeast GCN5 protein, was the first nuclear histone acetylase to have been identified. Okay. And very interestingly, the yeast GCN5 was found to exist in at least two distinct multi-protein complexes. One is called as ADA and another is called SAGA. I will discuss these multi-protein complexes in detail in a couple of lectures down the line, and neither of these, ADA or SAGA, is tightly associated with either TF2D or polymerase II, indicating that this multi-protein complex containing a histone acetyltransferase is another important player in gene regulation, in addition to other multi-protein complexes like TF2D and RNA polymerase II. Now remember, already we told in the beginning that transcription initiation requires at least 50 to 60 proteins, which consists of RNA polymerase and subunits, general transcription factors, and each one of the general transcription factors in turn are multi-subunit complexes. Now we are bringing another player into the field, another multi-subunit complex such as SAGA and ADA, which actually contain histone transferase or HATs as one of their components. So the number of proteins involved in gene regulation is increasing. And it soon became clear there are actually two types of HATs. There are two types of histone acetyltransferases. One is called HAT A, another is called HAT B. Now the A type HATs actually catalyze transcription related acetylations, so they have very important role in the regulation of gene expression. The B types HATs are actually cytoplasmic, and they actually catalyze acetylations linked to transport of newly synthesized histones from cytoplasm to the nuclei. So newly synthesized histones, they get acetylated and then transported to the nucleus by B type HATs, whereas the A type TAPs actually are staying in the nucleus, and they are involved in the modification or acetylation of histones at the chromatin. And so the observation that a transcription factor like GCN5P actually contains a histone acetyltransferase activity opened a Pandora box. So a number of people who had actually purified a number of transcription factors, co-activators and so on and so forth started looking in and see whether any of these transcription factors have histone acetyltransferase activity. And very surprisingly, they found many of the transcription factors or co-activators that they have identified in fact are capable of acetylating histones. So this opened up what is called as a HAT family of proteins, some of which I have mentioned here, like the GNAT family, which consists of GCN5 related N-acetyltransferases, and you have what is called the MIST family, which consists of MOS, YBF2, SAS2, TIP60, etc. There is another important, this is co-activator called CBP or P300. Again, we will discuss this in detail in next few classes. Many of the general transcription factors, especially the TBP associated proteins, also were found to contain histone acetyltransferase activity, and many co-activators which are actually associated with nuclear receptors like SRC1 and ACTR also were shown to possess histone acetyltransferase activity. So this linking or this convergence of histone modification or histone acetylation to transcription regulation is one of the important discoveries in the area of gene regulation. So many transcription factors were actually found to contain histone acetyltransferase activity, and this led to a very new line of thinking on regulation of gene expression. Now, if the histones are getting acetylated, there also have to be guys who will remove the acetyl groups from the histones. Only then regulation can take place, right? When acetylation is happening on histones and genes have to be activated, and then unless you remove the acetyl group and inactivate this histone acetyltransferase activity, gene expression transcription activation will go on and on, which is not good for the cell. So if there are proteins which are adding an acetyl group to histones, there have to be proteins which are removing the acetyl group from the histones. And soon people recognized there are in fact enzymes called histone deacetylases, or abbreviated as HDACs, which actually are capable of removing the acetyl groups added by the HATs. So these are called as HDACs or histone deacetylases. So you can see here you have histone molecules, and there are enzymes called HATs which actually add a acetyl group to the histones, and these acetylated histones are actually then deacetylated by a group of enzymes called HDACs, and there is an equilibrium between the HDAC activity and HAT activity at any given time inside the cell. So HATs such as GCN5P are actually found to be present in huge multi-protein complexes. Remember, one of the important functions of these HATs, they have to be associated with promoter regions or components of transcriptional machinery. So many of these HATs or the HDACs were found to be multi-subunit complexes, and they are actually component of other proteins which together form a huge multi-protein complexes. And as I told you before, these multi-protein complexes are different from those we have already studied, such as TF2D or RNA polymerase 2, etc., saying that there is another distinct entity of multiprotein complexes in which the HATs or HDACs are an integral component. For example, a multiprotein complex called SAGA actually contains SPT, ADA, GCN5 acetyltransferase, which the GCN5 is the acetyltransferase. And similarly, the human GCN5P was also found to be in a similar complex called STAGA. What the point I am trying to tell you is that many of these histone acetyltransferases exist as multiprotein complexes with other important regulatory proteins which play a very important role in regulation of gene expression. So, these multiprotein complexes, in which HATs are one of the components, they were found to interact with both TBP on one hand and acidic activators on the other hand. See, this is the reason I did not bring coactivators into the picture, because many of these coactivators whose job is to link the transcriptional activators on one hand and the general transcription machinery on the other hand, and many of these coactivators like the SAGA complex actually found to be histone acetyltransferase activity, and they on one hand are interacting with transcriptional activators, on the other hand they are interacting with components of the basal transcription machinery. So, there are many such proteins like PCAF, TFTC, which actually stands for TBP free TAF containing complex, were found to be members of this GNAD family of histone acetyltransferases. Now, very interestingly, the free GCN5 protein, when it is not present as a multiprotein complex, when it is on its own, can acetylate only free histones. It cannot acetylate histones which are bound to DNA in the form of chromatin, but as a part of the SAGA complex, it could actually acetylate histones. So, now we can see the significance of these HATs present as a multiprotein complexes, and these multiprotein complexes gives the HAT the ability to acetylate histones bound to chromatin, whereas the free GCN5 can only acetylate free histones. So, people soon realized that many proteins which were actually identified as coactivators, that is, these proteins are protein complexes which act as a bridge between transcriptional activators and general transcription factors. They all contain HAT activity, and I have listed some of the examples here, like human TAF 250, which is one of the subunits of the TF2D, and you have TAF 230, TAF 180, these are all TBP associated factors. They all turned out to be histone acetyltransferases. P300 CBP, a very important coactivator for many transcription factors, we will discuss in detail in later classes. And very importantly, when you knock out some of these HATs in mice, and these mice developed several developmental defects, clearly indicating that the histone acetyltransferases are playing a very important role in regulation of gene expression during development. And if you knock off the HAT activity, or you can knock off the genes encoding the HATs, you end up with many developmental abnormalities, and many of them are actually lethal. So, the embryos do not even develop at all, indicating that histone acetylation plays a very important role in developmental regulation of gene expression. And these HATs actually had the capability to acetylate specific lysine residues in H3 and H4. As I told you, histones are basic proteins, and each of these H3 or H4 histone contains a number of lysine or arginine residues. And interestingly, many of these HATs, when I mentioned some of these HATs, each of them HATs recognize a specific lysine residue or a specific arginine residue in either H3 or H4. So, these are highly specific acetylation reactions involving select lysine or arginine residues of H3 or H4. So, you can now see a concept is now emerging. What I have told so far, in order for transcription initiation or activation to take place, first the core promoter region, the general transcription machinery has to be assembled, then you get basal level of transcription. Then I told you transcription factors go and bind to upstream sequences, and somehow the binding of transcription factors upstream region can facilitate faster recruitment of this basal transcription machinery promoter, therefore promoting multiple rounds of transcriptionization and thereby enhancing the rate of transcription. Now we can realize one of the mechanisms by which these transcription factors are actually acting is by recruiting the histone acetyltransferases, which are actually part of many coactivator complexes like SAGA and so forth. So, the main purpose or main mechanism by which these transcription factors are enhancing the rate of transcription is actually by recruiting these HATs or multiprotein complex containing HATs, and these HATs in turn interact with some of the components of the general transcription machinery, and that is how the transcription factor function is now linked to the base general transcription machinery. So, activation or repression of transcription is determined by the equilibrium between HATs and HDACs. Okay. So, when you have acetylation, there is an enhanced expression of genes. When there is deacetylation, there is a decrease in gene expression, and there is an equilibrium between the HAT activity and HDAC activity at any given time at any given promoter region. And it was estimated that on an average 13 of the 30 lysine residues in a histone octamer are actually acetylated at any given time, and this steady state level of acetation is maintained by the opposing actions of HAT and HDAC complex. So, there is a constant acetylation of histone residues, lysine residues or origin residues of histones, and they are also getting deacetylated by HDACs, and this acetylation deacetylation is a very dynamic process, and this equilibrium is what ultimately is involved in the regulation of gene expression. So, many coactivators possess specific domains known as bromodomains, which can recognize the acetylated residues of histones. For example, in this case I have shown in this cartoon how one of the components of TF2D, namely the TAF250, that is the TBP associated factor 250, through their bromodomains, on one hand they can recognize the acetylated histone H4 residues, on the other hand the TAF250 in association with other components of TF2D can also recognize the core factor, and therefore these two processes can be recognized, integrated, and that is how it brings about activation of transcription. So, histone acetylation, the acetylated histone H4 is recognized by the member of a general transcription factor complex through the bromodomains, and then in combination with RNA polymerase can bring about transcription activation. Now, what we will now discuss is that we will talk now about histone deacetylases. So, once the acetyl groups are added to the histone molecules, they also have to be removed, otherwise the genes will be turned on all the time. So, how histone deacetylation takes place and what kind of enzymes are actually involved in histone deacetylation and what is their role in the regulation of gene expression. As I mentioned, there is an equilibrium between histone acetyl transferase or HATs and histone deacetylases, and the equilibrium in the HATs and HDAC actually determines whether genes are in transcription active state or transcription inactive states. So, I have just shown is here that you have, for example, a lysine residue of a histone, and you have HAT molecules which now add a acetyl group to the lysine of a histone, and therefore it becomes an acetylated lysine, and you also have these HDACs or histone deacetylases which now again convert it back to a deacetylated form, and this should result in transcription repression. So, there are a number of histone deacetylases have been described in the literature, and the best described HDACs are members of a common family that includes the founding member of from humans, namely the HDAC, and its homolog in E is called RPD3. So, remember the HAT HDAC system functions as a key regulatory switch of gene expression. So, when HATs are active, histones are acetylated and you have activation of transcription. When HDACs are active, you have repression of gene expression, therefore genes are inactive. So, it is the HDAC HAT system functions as on off switch for regulation of gene expression in eukaryotes. Now, as I mentioned, there are a number of HDACs have been discovered in a various number of eukaryotes, and these have been classified into a number of families for the ease of study. For example, the human HDACs belong to 4 classes based on their homology to the yeast HDACs, and these are called the RPD class, HDA1 class, SIR2s, and HDAC11 class. Class 1, class 2, class 3, and class 4, and there are various members of these things. We will not talk about the SIR2 class of histone deacetylases not now. They again play a very important role, and they also have a very important role in aging and so on and so forth. We will discuss the role of SIR2s in a separate class at a later stage of this course, but today we will talk about only about other class of HDACs and what is the relevance for gene expression. Now, why are these HDACs very important? Now, when people started identifying these HDACs and then asked the question, where are these HDACs expressed? When you looked at the expression of these various HDACs in normal cells versus tumor cells, and they found many of these HDACs are actually overexpressed in a number of cancer tissues. For example, the HDAC1 is actually overexpressed in prostate cancers, gastric and colorectal cancers. Now, this is although is only correlation that tells me that in many of these cancers there is a very high HDAC activity. Similarly, the HDAC2 is overexpressed in number of colorectal and gastric cancers. HDAC3 is overexpressed in lung cancer and several solid tumors, whereas HDAC8, if you knock down, it inhibits the cell growth in several human tumor cells. So, these studies clearly told that the HDAC, the histone deacetylases, when they are active, they can actually may be involved in cell proliferation. So, what could be happening is that these HDACs may actually be deacylating the promoters of many tumor suppressor genes, and therefore the expression of these tumor suppressor genes may be blocked, and as a result the cell has become cancerous. And the evidence actually comes from here. In the case of HDAC8, which is actually expressed in the particular tumor cell line, when you knock out this HDAC tumor 8, you can actually inhibit the cell growth of, that means some of these tumor promoter genes expression is getting turned on, and therefore it is resulting in the cessation of growth, indicating that this HDAC plays a very important role in cell proliferation and cancer. Now, as is true for many of these HATs, the HDACs are also found in a number of multi-protein complexes. For example, I have given two examples here. There is something called as SYN3 co-repressor, and there are transcription repressors such as SMRT and MCOR. We have discussed all these things in previous classes, and HDAC1 is a very important component of some of these co-repressor complexes, and there are also many examples where many co-repressors and repressors which are actually on a negative regulation of transcription, they have been associated with histone deacetylases. Now, I want to now discuss two examples just to bring into you, get you an overall picture of how a particular signal transduction pathway will ultimately result in either activation or recruitment of a HAT leading to activation of transcription, or in the presence of HDAC can result in a repression of transcription. Now, in this example, I am going to tell you a particular signal transduction pathway which involves the generation of a second messenger called cyclic AMP. Now, there are certain growth factors. When these growth factors interact with some specific cell surface receptors, this growth factor receptor interaction ultimately results in the activation of an enzyme called adenylate cyclase. This adenylate cyclase converts ATP into a molecule called cyclic AMP. Now, cyclic AMP is a very important second messenger. I am not going to discuss all the important activities of cyclic AMP in this class, but remember, cyclic AMP is a very important second messenger and plays a very important role in the regulation of gene expression as well. Now, when cyclic AMP is not there in the cells, when cyclic AMP is not present, there is an enzyme called protein kinase A which is present in the cytoplasm of cells, and this enzyme has two regulatory subunits, two catalytic subunits. The regulatory subunits I have shown it in green, the catalytic subunit I have shown in red. Now, as long as this regulatory subunit is associated with the catalytic subunit, this protein kinase A remains in a inactive form. So, it cannot, is not enzymatically active, it cannot add phosphate group to a protein. Now, when a growth factor binds to a cell surface receptor and activates an adenylate cyclase, and one cyclic AMP is made inside the cells, now this cyclic AMP now comes and binds to the regulatory subunit of protein kinase A, and once the cyclic AMP binds to the regulatory subunit, it causes the dissociation of the regulatory subunit from the catalytic subunit. Once the catalytic subunit is dissociated from the regulatory subunit, this catalytic subunit now becomes active, it is catalytically active. Now, this protein kinase A now translocates from the cytoplasm to the nucleus. Now, this catalytic subunit phosphorylates a very important transcription factor called as CREB. Now, CREB stands for cyclic AMP response element binding protein. Why is it called? Because this protein actually goes and binds to specific sequences called cyclic AMP response elements, which are present in the promoters of genes which are activated in response to cyclic AMP. So, if there are certain genes which are getting activated in response to cyclic AMP, it means that these genes contain cyclic AMP response elements, and these cyclic AMP response elements needs to be bound by CREB. Now, what is important is that the catalytic subunit now goes and phosphorylates a specific serine residue of this CREB, and therefore the CREB now becomes phosphorylated. Now, this phosphorylated form of CREB now interacts with a histone acetyl transferase called CBP/P300, and as a result, this results in the activation of transcription from genes containing cyclic AMP response elements in their promoter. So, you can see here the binding of a growth factor to a growth factor receptor for the cell surface has come all the way, and how this signal is propagated all the way into the nucleus. Ultimately, it has resulted in the recruitment of a histone acetyl transferase leading to the activation of gene expression. So, if genes have to be activated in response to cyclic AMP, recruitment of histone acetyl transferase by phosphorylated CREB is a very important step. So, this is how a signal transduction pathway operates. This is just one example. Now, I will give you another example just to show how a transcription is shut off by another signal transduction pathway, this time involving a histone deacetylase or a HDAC. Now, histone, we have discussed, for example, steroid hormone receptors. Now, I told you steroid hormone receptors belong to a family called nuclear receptor superfamily, and they contain a zinc finger DNA binding domain. They usually contain what is called a DNA binding domain, through which they go and bind to specific DNA sequences in the promoter regions, and they also contain a ligand binding domain or a transcription activation domain. Just like in the case of glucocorticoid receptor, when a glucocorticoid hormone binds to the ligand binding glucocorticoid receptor, it results in the activation of transient glucocorticoid response genes. In the case of thyroid hormone receptor, the ligand binding domain binds to thyroid hormone. Now, when thyroid hormone is not present, that is, if you do not treat cells with thyroid hormone, the thyroid hormone receptor which is present in the nucleus interacts with the thyroid hormone response elements in the promoter regions of genes, and this thyroid hormone receptor, in the absence of the ligand, this ligand binding domain interacts with a co-repressor complex which contains a histone deacetylase, and as a result these genes are not transcriptionally active. What I am trying to say is that when there is no thyroid hormone inside the cells, the thyroid hormone negatively regulates the expression of genes responsible to thyroid hormone, because it interacts with the histone deacetylase. Now, the moment you treat the cells with thyroid hormone enters the cells, because thyroid hormone is a very small molecule, it easily enters the cell and it goes and binds to the ligand binding domain of the thyroid hormone receptor, and this binding of the thyroid hormone to the ligand binding domain receptor, which I have shown here in this triangle, now cause a conformational change in the ligand binding domain, and as a result the ligand binding domain of thyroid hormone receptor can no longer bind to the co-repressor complex, and therefore the histone deacetylase cannot interact, therefore no deacetylation of histone takes place, and therefore now genes are activated. So, in the previous example I described to you how cyclic AMP results in the recruitment of histone acetyltransferase leading to activation of transcription. In this case I told you how the addition of a small molecule thyroid hormone results in the dissociation of a histone deacetylase, resulting in the activation of transcription. That is a positive regulation of gene expression. This is an example of a negative regulation of a gene expression. In one case there is a HAT recruitment, in another case there is a dissociation of HDAC from a transcriptional activator. So, this is how various signal transduction pathways are integrated. Ultimately, the activation of genes ultimately involves either recruitment of a histone acetylase or a histone kinase or a histone methyl transferase or histone deacetylase, depending upon what kind of genes we are studying. Now, what is very important I want to now describe is that people are now started identifying a number of small molecules which can go and inhibit these various HDACs. I have listed some of these compounds here, like sodium butyrate, trichostatin A, superolide hydroxymic acid, or abbreviated as SAHA, valproic acid, and so on. And when you treat cells with various HDAC inhibitors, they induce a number of important processes, like for example they can induce cell cycle arrest, they can cause differentiation of cells, they induce apoptosis in colon cancer cell lines, and so on and so forth. And many of these compounds which are HDAC inhibitors have found to have anti-cancer properties. I will only give one example because it has already entered clinical trials. This particular example called SAHA, which is a very potent inhibitor of HDACs, is actually now commercially available as Zolinza as a trade name, and Zolinza was actually approved on October 6, 2006 by United States Food and Drug Administration for the treatment of a specific type of skin cancer called cutaneous T-cell lymphoma and Sézari's disease. You can actually get all the details if you actually click onto this website. So, here is an example where an inhibitor of a histone deacetylase is now being used for treatment of a specific type of cancer, clearly indicating that this HDAC inhibition of the activation of this HDAC probably is resulting in the activation of certain tumor suppressor genes, and that is how it is causing cessation of cell proliferation. And here you can actually see how basic research understanding gene regulation can translate in the form of benefits. Here is a compound which inhibits a histone deacetylase, and this has a potential as used as an anti-cancer agent. Now, the other important point I would like to discuss is that in addition to the deacetylation of histones, many of these HDACs, they also go and deacetylate many other transcription factors such as P53, U2F, and many other factors. And in these cases, just as deacetylation of histones results in tighter binding to the DNA, deacetylation of some of these factors results in either reduced DNA binding or reduced transcription activities. I have just given here a list of a number of proteins which are not histones, but they are all substrates for HDACs. The point I am trying to make is that in addition to histones, there are many non-histone proteins which are also substrates for HDACs, and they also have a very important function because it either results in the reduced DNA binding or reduced transcription activation. So, the take home message I am giving you from all these studies is that here is a cartoon which tells you when transcriptional activators recruit histone acetyltransferase or HATs, it results in histone acetylation and transcriptional activation. This is a general principle we have talked so far, whereas when transcription repressors recruit HDACs which remove the acylated groups from the histones, it results in tighter binding of the histones, therefore results in inactive chromatin and therefore transcription repression. This is the take home message from the discussion we have had so far. HATs means transcription activation, recruitments of HDAC means transcriptional repression. This is the take home message. So what I have given in these next few slides is that I have actually discussed or listed a number of important papers which have played a very important role in understanding the role of histone acetylation or histone deacetylation in the regulation of gene expression. So you can go through some of these papers which are, I would say, are original research articles and have played a very important role in understanding the role of histone acetylation and histone deacetylation in the regulation of gene expression. You can just go through. There is also very nice review article in Nature which talks about the language of covalent histone modifications and very briefly describes how the covalent modification of histones affect regulation of gene expressions, and I have also listed 3 papers which discuss very nicely about 3 important points, namely chromatin structure and modification cannot be viewed as a process that is independent of transcription initiation. Remember in the beginning I told you, before the discovery of relationship between histone modification and transcription regulation, people who are studying chromatin structure were different, people who are studying gene regulation were different, but once people have realized that chromatin structure plays a very important role in gene regulation, these two people started coming together and people started understanding how the chromatin structure is influencing gene regulation, and these papers very nicely discuss some of these aspects. For example, chromatin structure, chromatin is not simply a structure that serves to compact DNA in the nucleus, but it also has a very important role in transcription activation, and histone acetylase and histone deacetylases provide a critical link between chromatin structure and transcriptional output. So in addition to chromatin structure, histones also play a very important role in the regulation of gene expression, and today we have discussed 2 important modifications of histones, namely histone acetylation and histone deacetylation, and now histone acetylation by HATs results in activation of transcription and deacetylation of histone by histone deacetylases results in inhibition of transcription. And I also gave a one very important example where a compound called SAHA, which is a small molecule inhibitor of HDAC, has already been approved as an anti-cancer drug for treatment of a specific type of cancer. Okay. So we clearly indicating that understanding the mechanism of gene regulation, understanding the mechanism by which histones either activate or repress gene expression, and how acetylation or deacetylation of histones can result in activation or repression of gene expression has very profound implications in a number of disease processes including cancer. And at this time I am going to talk mostly about the basic aspects of gene regulation. We will come back later at the later stages of our course and discuss in more detail how some of the basic knowledge that we have gained from understanding the basic aspects of gene regulation has been translated or is being translated in the form of specific therapies for a number of disorders including cancer and so on and so forth. I think I will stop here.